You're gonna do that right here by the thing. Man, you are right behind me. I'm really hoping I definitely clicked on the right link on Zoom today, so I'm just hoping that it's like gonna work on here since nobody is showing up, and that worries me. Don't start. Awesome. A person joined. You have no idea how happy I am that this is working. I'm waiting a little bit for some more people to join. Hi, everybody. We're just waiting on a few more people to join and we'll get started. Just wait maybe 1 or 2 minutes. Just for stragglers. I know not a lot of people are going to join, but that's okay. Y'all, I about turned my TV on to put something on. I'm gonna watch it in the background. I'm going to do that. It just like was out of habit. I'm sitting in the living room. My neighbors are installing a new roof, and so my office is right by that side of the house, and so I didn't want to be in my office, so we're relocated to the living room. All right, so I'm gonna go ahead and I'll pull up the PowerPoint stuff now. Still sharing as it is. And let's get started. Let me pull down my, this of course has to be right by the record button. Why wouldn't it be? Okay. So this is week 3 review, but we really only went over one chapter this week, right? Because we were only here on Wednesday and Thursday. So we're just going to be talking about that one chapter, which is going to be chapter 7, but it is virology, it is the viruses, and I know it's a really new concept, especially compared to, eukaryote cells, which is what our cells are. Most of us are familiar with those, or even bacteria, and the bacteria are going to be, maybe a little new for us, but at least we know, had a concept of their cells. I mean, they're alive and everything like that. So viruses are a completely new concept. Before we go on, I'm just going to remind you guys of that little layout that we had going on previously with the cells and everything like that. So like if we were talking about, I guess let's do like microbes. And those can be divided into 2 groups, which is the acellular and the cellular. We were previously learning about the cellular guys, which can be divided into the, excuse me, eukaryotes, and then the akaryotes. And remember, this is going to be, they have a nucleus, and then they don't have one. And then we had originally divided up the akaryotes as, we talked about archaea being a thing. We didn't really get into them too much. They're just the ones that tend to be the extremophiles. So archaea, that laundry, scared spaghetti. Okay, and then bacteria. And we didn't really even go into the differences too much about these guys, except for the fact that I'll just remind you that the archaea are just really different. They look similar under the microscope. They're similar size. They're unicellular. They can live in a lot of different environments, but their makeup is completely different. Their lipids, like we talk all the time about phospholipids making up our membranes. Archaea don't even have phospholipids. And as far as their genetic material goes, they do have DNA, but their DNA matches ours more closely than it does bacteria, and their ribosomes look more like ours, and it's just strange. So that's why we didn't really go over the details too much with those. We don't need to know a lot about them, and they're really different, like a lot more than you might think. So that's how we kind of looked at those guys. And with the eukaryotes, the guys that have the nucleus, we even broke these down in further categories. We looked at the, let's see, I'm trying to think how we started with this. We looked at the fungi. And then we looked at the protozoa. This email. Oh, that's my auto email. Yeah, it's the auto email. And we looked at Animalia. I should say protozoa falls into protista. So if I was being, good, I would say that falls into that. So let's amend this, if you will. Protista. All right. And yeah, there's other groups within, kingdom that within eukaryotes, right? So we talked about plants and that, but like this is what we're talking about in microbiology. So in the fungi, we even had that broken down into yeasts and molds, what we're concerned about in microbiology. In the protista, we had broken those down into the algae, which don't cause any disease, but they were one of the groups, right? The subkingdom algae, and another subkingdom of protozoa. Which are always going to be single cell. All the protista are single cell. And then the Animalia, the ones that we were concerned about, really, of course, we want to know about us because we fall into that category and we're going to be the hosts for these diseases. But for the diseases themselves, we really talked about the helminths, which are the parasitic worms. Wish I hadn't overlapped, but that's okay. So that's kind of how we're going to divide those up. But then the acellular guys, all on their own, they get their own little deal. They are not made of cells. They cannot divide on their own. They don't have everything that they need to make new copies of themselves. They're going to have to hijack all the machinery from the cells that they're infecting. So that's why they're not true cells. That's why we don't know that we can say that they're living or not, and why we just go ahead and just say active or inactive. So yeah, you can even, like, if I were to even justify this, because we're going to get into that in this. Let's say, so the acellular guys are not truly living. So I'll just put not living. If your opinion is that you still think that they're living, that's fine. I'm not going to test you on that, but I want you to be aware of the fact that there are many that are, of that mindset. So of course, we had two things that we learned about here, both of which are in chapter 7, and that's the viruses and the prions. Prions are just misfolded proteins only. They do not have nucleic acid. They don't have genomes. They're literally just a misfolded protein that causes other proteins near it to also misfold. Proteins of the same kind, I should say. So if you had a protein that was an enzyme that made ATP, if it is misfolded, then it will go near other enzymes that also made ATP in the cell and potentially near to cells that are nearby and change them so they are not folded correctly either. And so that's what it will do. Those misfolded proteins no longer perform their normal function. And they also, because they're misfolded, aren't going to interact with their environment the normal way that they're supposed to. So they will aggregate together and create what we call plaques in the brain. We hear about this a little bit in Alzheimer's. It happens just naturally in Alzheimer's, but with this is a propagating effect. So, but yeah, it'll propagate like that, just getting near normal protein and turning it into misfolded protein. That'll then aggregate and cause holes in the brain, which is where we got the spongiform from. And remember, that's spongiform encephalopathy. So that's the prions. That's really all we need to know about them, other than you might want to be familiar with some of the diseases, what they're called. Especially, I would say number one is Creutzfeldt-Jakob disease. I will never make you spell it. Okay, it's never gonna have you spell it. Creutzfeldt-Jakob disease, and that is CJD. The Jakob is with a J. That's like a German Jakob, like Jacob. But anyways, so CJD is the human version, essentially, of the naturally occurring, not eating other people and not having, genetic insomnia or anything like that. CJD is the typical version of that. So that's prions really in a nutshell. And then we had the viruses. Remember, we had talked about viruses consisting of the ones that could be naked or enveloped. If you want to, you could also divide them by the different kinds of genomes that they have. When we're classifying the viruses, typically you're going to classify them based on their genome first, but I know that there's a slide about classifying them, but just see it as we're classifying them by their differences, whether they have these different kinds of genomes, whether they're naked or not, and then eventually like the literal genetic differences between each one that falls into each category, and even like what organisms they can infect and what diseases that they can cause, and if they need a vector to cause disease first. And I know you guys know about some of the diseases that have to have a vector in order to be transmitted, or that typically require a vector in order to be transmitted, in which case we are talking about, sorry, I get emails and everybody on the faculty senate wants to send out their info on Fridays, I guess. But anyways, you okay, Meatball? You sure? Okay. Sorry. I guess what I'm trying to say is focusing on the differences to categorize these guys. Some of these require vectors, things like the ones that are transmitted by ticks or mosquitoes and stuff like that. You know, that's how they're typically going to be transmitted. Like dengue fever is transmitted by mosquitoes, or West Nile is one that we're maybe more familiar with here in the United States, and Zika. Can you potentially get these viruses if you get a blood transfusion that has these viruses in them? Yeah, you can, but that's not usually how they're transmitted, right? So we can even talk about them that way, just how they're transmitted. So that's our little breakdown, a little reminder, if you will, of everything that we've kind of gone up to at this point. And I really wanted to, spread it out and look at it again like that, so that we could look at the big picture of all the stuff that we have learned. And can we break down the bacteria into something like, we go back over to bacteria, which is over here, by the way. Can we break that down? Well, of course we can. We could break it down into gram positive. I didn't know that was going to go off the screen, or gram negative, and all sorts of stuff like that. And so we can get even more in detail, of course, but this is our general concept of how we can break down everything that we've just learned about for the entirety of Unit 1 in one big picture. And when I tell you guys that one of the things that you guys can do to help study is, creating mind maps or flow charts, or, writing your own outlines or your summaries about certain topics or chapters or whatever. This is what I'm talking about, just kind of breaking it down. So you could even start with a basic outline like this, and then say, okay, well, let's look more into, the gram negatives, what makes them gram negative, and then make a little chart even on its own, or build off of the chart you already have, which is easier to do if you're doing this on like an actual like note app or something on your iPad and everything. You can just make it all in one big screen. So that's how I recommend that you guys kind of go over this stuff, really break it down and get into the nitty-gritty details of all the stuff that we've learned with all of the terms that we have on the study sheet, and gone over that, and make sure you can see it in the big picture. That's going to help a great deal, as well as even getting you down to the details that you need to understand. It kind of puts into perspective the importance of these differences that we've learned about in these microbes. So I want to get that in before we get into reviewing Chapter 7's, commonly misconceptions. So again, like usual, we will start off with our agenda with Chapter 7, and then we'll go into the missed topics, since we're only doing one chapter here. So for Chapter 7, remember we're talking about the viruses and the prions, but the acellular microorganisms. So viruses are obligate intracellular parasites. And, they're simple, they're non-cellular, they lack organelles, and as simple as they are, they seem to cause devastating diseases. Other concepts that we talk about in this chapter: a single working virion. And a virion is a single virus particle that is like actively capable of infection and stuff like that. So it's always going to have a genome. It could be single-stranded or double-stranded, DNA, RNA, positive or negative sense. It's going to be one of those. So there's a whole bunch of options that we have, and those can fall under both the DNA or the RNA. So double-stranded or single-stranded DNA, double-stranded or single-stranded RNA, and the single strands can be positive or negative sense for both of those. So there's a lot going on there for our possible genome types for the viruses. Not all viruses have DNA. I want to be clear about that. They either have the one or the other of these, never multiple. So if they are a DNA virus, they only have DNA. If they're a positive sense RNA virus, they're only going to have positive sense RNA in their little virus capsule. Okay, so they always have a capsid. So we went over genome and all the different kinds that they can have. They can also have a capsid. And I do want to clarify with those genomes. When I say these things, it can be abbreviated, double-stranded DNA. Or you could have single-stranded DNA. That can be positive sense. So the ones that you could read potentially if it was, you could read it if it was RNA right away. So, or the negative sense, single-stranded DNA. So it had to be make a copy of it before it could be, turned into a product. And then we can have the double-stranded RNA. And then the two different single-stranded versions of those. Keep in mind that something like double-stranded RNA, which I remembered to mention in one of my classes, was that double-stranded RNA is an immediate no-no for our bodies. If we see double-stranded RNA at all, it's an immediate sign that we've been invaded. We don't have double-stranded RNA in our cells. We have structures that we've built out of RNA, like the ribosomes or the transfer RNAs. We have single-stranded RNA, like messenger RNA. But if it is a double-stranded double helix of RNA, we don't have that in our cells. So our cells have adapted, our immune system has adapted to see that as an automatic trigger to attack. It is similar, as I'm sure you can imagine, we do the same thing to lipopolysaccharide or LPS or endotoxin, all of that the same thing for the outer membrane of the gram negatives. So we shouldn't have that hanging out in our, bloodstreams and stuff like that. So that's also an automatic call to arms for our immune system. So we don't even need antibodies or anything to attack that. We can just automatically see it as foreign and just start attacking right away. Yeah, we'll learn more about that in Unit 3, but understanding the importance of how strange this is to our body, I mean, it's kind of a big deal, So anyways, that is a pretty interesting aspect of these viruses. So moving on, we have the capsid. This is just the protein coating that is surrounding your nucleic acid genome. Remember that the genome, right up here, plus the capsid has a term for its own, and that's the nucleocapsid. That's one structure of a virus that all viruses will have, because all viruses have a genome and a capsid, whatever their genome is. So all viruses will have that. If you're a naked virus, then that's all you would have. And you have the spikes, of course, on the outside to help you get into your host cell, no matter what. So the spikes depends on what kind of virus you are, where they're going to go. If you're a naked virus, the spikes will attach to your nucleocapsid. If you're an envelope virus, then you're going to be having your nucleocapsid surrounded by that, lipid bilayer that you stole from your host cell when you budded out of it. And that's going to be studded with the spikes. So while you were generating all the parts of your virus, you were putting those spike proteins on the surface of your host cells. So you could just push your way through that membrane and coat yourself in that mess and have your spikes on you. So the spikes, remember, those are spikes. These are glycoproteins. So again, all that means, it's not a scary word. It just means proteins plus glyco. You should be feeling like that should be sugar, and you'd be right about that. So sugar protein. And that's the only importance of this, is that it's going to be the key that helps you, engage with the lock on the cell that you're trying to infect. And we've mentioned that that has a lot to do with, the host range, whatever, the lock that you're engaging with on the surface of those cells, that lock, you have the key to that lock only. And if that lock is on a whole bunch of different kinds of cells, you can infect a whole bunch of different kinds of cells. If it's only on liver cells of humans only, then you can only infect the liver cells of humans. So that can determine host range there. So that's how the glycoproteins and their importance. And like we said, some of them have that envelope that's a lipid membrane, the lipid bilayer that they're stealing from the host. Could be the cytoplasmic membrane, which that's what we have on the outside of our cells. So it could be that. Or if it's a DNA virus and it was being made in the nucleus, which is where all of the machinery is for making DNA, so it would have to be replicating in the nucleus, then they can bud out of the nuclear membrane instead. And it gets more complicated than that after that, but those are the two main ways that they can deal with it. So be aware of the steps of the viral life cycle here. Adsorption is just sticking to your host cell by that lock and key mechanism. You found the receptor on the outside of the cell, just a protein or something that your glycoprotein spike engaged with. Okay, that's adsorption. Then we have penetration and uncoating. Those usually happen kind of in steps together. Penetration is getting taken into the cell somehow. You could be merging with the surface of the cell. Like if you have a membrane, so that could just like incorporate as you like your membrane gets docked up against the membrane of your host, and they kind of merge together, and that sort of just empties its contents, which are the nucleocapsid, into the host cell, and then things go from there. So we can have that going, or we can endocytose. That means you come and you hit up with your docking area on your cell, and the cell is then triggered to engulf you as it would want to do. It would be a normal mechanism of the cell, and it would engulf you and swallow you up into a vesicle, and then it could break down your parts and then release your genome. So there's two different ways you can penetrate and then uncoat. Uncoating just means that we're releasing the genome. Usually going to be breaking down the proteins by normal enzymes in the cell. It's not going to be anything specific or horribly nasty or anything like that. The cell usually doesn't know that it's infected until it has gotten into replicating the virus. Usually. So, especially if it's a first-time infection. Then we go through synthesis. That just means making copies of the proteins that we need to build the new nucleocapsid and the spikes. And we need to make copies of our genome. Remember, the genomes of the viruses can exist in segments, or they could be one whole big long genome. So they can have variety there. So we're going to synthesize the copies of our genome, and then we're going to assemble our new, our new virus particles, our new virions, and have them bud out through the membrane if they need an envelope, or they can bud out. So that's one way for release. They can bud. So that's if they're stealing the envelope. If they're not stealing the envelope, so in the case of naked viruses, they're going to be exocytosed. So exocytosis. The cell is just going to get rid of them. They're going to put them in vesicles and just kick them out of the cell, like they would waste or any sort of protein or signaling molecule that they wanted to get out of the cell normally. That's how they're going to get rid of these newly made viruses, so that they can go on and infect new cells. So that's typically how you will see that with the, again, the enveloped or the naked. All right, so cytopathic effects. These are negative changes in the cells that are going to be caused on the cells by a virus. It's usually going to be things that actually are causing, say, end stage sort of effects on the cell. So visible changes in the cell shape, or visible changes in that cell that is infected. So if you're infecting a cell and you're affecting the genes or whatever in that cell, that itself is not a cytopathic effect, okay? Because that's going to affect the cells maybe down the line, but not that individual cell. It's not necessarily being negatively infected itself. So we want to, in cytopathic effects, we're talking about the negative effects of that infected cell. Is it being enlarged? Is it having a weird shape? Is it becoming disoriented on where it's supposed to be bound to, or its orientation as far as like how it's facing, maybe the bloodstream versus the inner tissue or something like that. So the syncytia development, remember that is whenever we have cells merging together much like we might have the viruses merging with the cells initially, because it's triggered by the viral proteins on the surface of the cell. It's like if you're a cell, like here's a cell, right? And you've been coated with all these spike proteins, right? As you're making new virus inside of you. And you've got a new cell next to you, and it's got all of the locks that those spikes are keys for. And it's just chilling on here. Well, then these can engage with those and then cause these membranes to start to merge together. And then they will become one eventually, quite literally. And there we go. And then so this one would have a nucleus, this one would have a nucleus with its little DNA in it of the cell itself. And then now we've got a multinucleated massive cell that happens because of that interaction to the cells nearby. And it also leads to the cell nearby becoming infected if it wasn't already. So all of this can be happening. Those are syncytia. Let's see where that term, I don't know if it's on here, but yeah, so that's called. Just so you are aware of that, if I were to ever say, are syncytia a type of cytopathic effect? That's not a weird term to you know, okay? So that's cytopathic effects, bad, visible, major changes in the cells that you see as a result of viral infection. All right. Bacteriophages. These are just viruses that infect bacteria. They infect only one kind of bacteria, only one species only. So if it's a bacteriophage, a virus, again, that infects bacteria, that only infects, E. coli, then it will only ever infect E. coli. And that becomes important later on when we're talking about some diagnostic procedures that we're going to be using. And I think that's chapter 13. No, chapter 18. One of those, 13 or 18, I get them confused. So I think it's 18 now that I think about it. But it doesn't, we'll have to know about it, is what I'm saying. So that's how the bacteriophages kind of will work. They'll come and they'll bind on to the surface of the cell, just like the viruses that infect our cells do, the ones we've been talking about. But they'll just come and recognize the little docking on the bacteria, but they don't go into the bacteria. They just inject their, genome, not their nucleocapsid, because all they are is a nucleocapsid. They don't have envelopes. So they're a nucleocapsid, a naked virus with these crazy shapes that look like spaceships, coming over and recognizing that this is E. coli, squatting down and injecting their genome straight into the E. coli cell. The E. coli cell, because they don't have a nucleus or anything, they can start working on that right away. So they'll start, oh yeah, here comes some more, DNA, or here comes some RNA, whatever it is, and start interacting with it immediately to start making products to make the new bacteriophages inside of their bacterial cells. They'll make new phages and, their proteins and their genomes and whatever like that. And then they will literally keep doing that until they bust out of the cell, because there's so many of them that have been made. And that's called lysis. We talked about lysis being breaking open of a cell. So they will lyse the bacteriophage. The bacteriophages will lyse the bacterial cells. They will never bud out. Okay. Okay. So this whole thing, this whole situation is called the lytic cycle. When we have that normal infection process going on, normal replication, lysis cycle, that's called the lytic phase of the lytic cycle. So that's lytic. That's the normal way that we've been thinking about viruses this whole time. We also can have lysogeny. In lysogeny, this is whenever temperate, and that's another word that we need to be familiar with, temperate bacteriophages, which can switch between these two. Temperate bacteriophages can switch between lytic and lysogeny. Lysogeny is whenever we are incorporating the genome. So the genome is going to incorporate of the virus, to be clear. The viral genome is going to incorporate itself into the bacterial genome. And then it just kind of waits out and hangs out there. And as the bacteria just normally divide, because they're not bursting open, they're not making virus or getting killed by, virus multiplying and bursting them open. They're just living with this thing in their genes that they don't really know about or care about. And when that is happening, when the bacteria divide, which they do quite often, maybe not as often as viruses can build themselves, but the bacteria is still moving pretty quick. They will be making copies of those genes in every single daughter cell that they generate. So that's how they're passing on that virus, so to speak, without having to have the virus being built up. So that's how that's going to work. So they can switch the temperate bacteriophages are capable of switching between the lytic phase, where they're making viruses and bursting out, or the lysogenic phase, where they are, incorporating into bacterial cell and just having bacteria copy the genes. And they can get triggered to come back out again and start going back into the lytic phase. That's going to be a process known as induction. So we're inducing, like you're inducing labor. So that shouldn't be a crazy term either. So it's easy to imagine that would be what was happening if we're switching back and forth. Because like I said, if they can switch back and forth, they can switch back and forth, right? So yeah, so that's what our temperate phages can do. Sometimes whenever you are incorporating your viral genome, your phage genome into the bacteria that you're infecting, you can be bringing with you genes. They can be genes that are always brought with you because of how you normally infect other bacteria. They could be genes that you just randomly picked up because you randomly insert into parts of bacteria and you just like took that part with you randomly. And they could be inserted in specific places of the genome or in random places of the genome. And there's different ways that can happen. But either way, if you are inserting a new gene into a bacteria through this lysogenic cycle, so inserting the genes into your bacteria, and they're along with that, while you're doing that with your little phage genome, you've carried with you genes from a previous cell that you came from. And that can impart those extra genes into the new host bacterial cell. And if those genes are beneficial in some way to the bacteria, like creating a toxin or some sort of resistance, then that is called lysogenic conversion. And that is here on here, right here. So lysogenic conversion is the term for that, getting new genes through that lysogeny process, that lysogenic phase. And I remember the difference between lytic and lysogenic, just so that we're on the same page here. I know I said it before, but to remind you guys that because lysogenic has gen in it, like gene, that's how I remember that's one that involves the genes and inserting into the genes and maybe bringing in new genes sometimes. The lytic phase is just where you're going to create viruses until you lyse the cell. That's all that it does. So moving on, we have the proviruses. The provirus is in eukaryote genomes. Whenever viruses insert their genes into our genomes, like we said with herpes viruses or HIV, when you see this happening, that gene, the virus that has been inserted in there, its genome is now called the provirus. So you're carrying the provirus. It's the genes that would code for an entire new virus, but it isn't because it's just hiding out in your genes. So that's why we call it the provirus. It's not quite a virus yet, just the genes for it. So this can make viral infections become latent. And you've heard that term before, which means you got an infection and now you're just hiding it out, whether that be chickenpox or herpes or HIV, you still have it in your genes. That is the same thing for the bacteriophages, because we said that we just talked about lysogeny and how they did that. But they are not called proviruses, they're called prophages when they go through that lysogeny. And lysogeny and the lysogenic phase versus the lytic phase, that's only going to be in bacteriophages, not in eukaryote. Okay, those are not terms that we use for eukaryotes. Eukaryotes get provirus and they have latency and stuff like that. So they can switch back and forth, but they are not called lysogenic or lytic cycles. Oncoviruses are viruses that when they do this with you, they create a provirus. engage with your DNA typically, they're affecting how your cells are supposed to be replicating and can lead to improper replication of those cells. And that leads to the development of cancer. That's called the transformation of those cells. That includes things like HPV. And lastly, the last thing we talked about in class were the prions. I know we talked about interferon too, right? And that's the guy with the shotguns and calling your neighbor up and making sure your neighbor knows that bad guy's coming so that he can get ready for it, right? That's interferon. It's the signal sent to the cell next door to remind you that, to let you know that the bad guy's coming your way so you can prepare ahead of time. And cells will do that if they get that signal. So again, interferon, because we're interfering with the bad guys, the viruses, ideas. So that's this, interferon. It's abbreviated as IFN. There's a whole bunch of different kinds of them, but that's one we're talking about now. So anyways, moving on to the prions. The prions are literally just misfolded proteins, like I said previously. They do not have a genome of any kind. There, in fact, doesn't seem to be any sort of mutation or gene that makes you predisposed to getting a prion. There doesn't seem to be any relation at all, but it can somehow still seem to be heritable. And we don't know why that is. We think that's probably dealing with other mechanisms that are involved in helping that protein, misfold, if you will, that we're not looking at, like certain genes that we haven't quite investigated yet. But that's a really complicated question. So we don't really know where they have come from originally, but we know that once you have the misfolded protein in your system, it can cause your normal proteins to misfold. So specifically, like I said, it would be one protein that is a normal protein. If that one kind of protein misfolds, then it causes others of that same exact protein to misfold. And they have to come into basically contact with one another. So it's like a cascade effect, if you will. But yeah, once that happens, they will build up and cause plaques in the brain tissue. It's typically going to be neurologically related and not in any other tissues. You can find it in other tissues in the body, but it's usually going to be nervous tissue that is the biggest issue. So these will cause spongiform encephalopathies. And again, that would include Creutzfeldt-Jakob disease, CJD, bovine spongiform encephalopathy, which is BSE, or mad cow disease. We also have scrapie, which is like the most studied one in sheep, because it's easier to study sheep than these other organisms. There's chronic wasting disease in deer, which you should be testing for if you are a deer hunter. They really want to follow and track the spread of this prion disease because it can be highly contagious between deer. It shouldn't affect people though. Then fatal familial insomnia is one that's really like super understudied because we just don't have, it's super rare. And it's a little bit different from the other ones. And then we have Kuru, which was the, tribes, the cannibalistic tribes, venerating their dead and all that, spreading Kuru that way. So that is the gist of chapter 7. I wanted to spend a little bit more time reviewing this one because it is a lot of new information. All right, let me see if I want to check to see anything came up. It doesn't look like it. It looks like we're good. I don't know why it doesn't have, whatever, it doesn't matter. Let's go back. All right, next. So now let's talk about the commonly missed topics. I think there's 3 or 4. It shouldn't take too long now that we've gone over the gist of a lot of this information. So chapter 7, again, viruses. All right, so our most missed concepts. The first one is going to be the cytopathic effects. Remember, these are going to be structural and functional changes that happen in that host cell when it is infected. This doesn't include things like passing on the genes to later generations and them having, replication issues. It talks about the host cell that is infected and that infection causing these issues. They're usually going to be, like I said, structural and functional changes that you can see sometimes in a microscope, even when you're studying viral infections. So you can look at your cells and say, yep, these definitely got infected when you're studying them. They also affect the health of people that are being infected. These happen when viruses infect the cell, take over the machinery to replicate itself, and that will disrupt the normal cellular functions and cause various types of damage, which we kind of talked about a little bit. But some of the types of cytopathic effects that you may see: cell lysis. Some viruses can cause cells to burst open. We typically don't see that in a lot of our eukaryotic cells, but it can happen. There are some viral infections that will burst out of your cells. And that is called lysis when it happens in our cells, just like it would if it was happening in the bacteria, which is what always happens in the bacteria. That will release viral particles, and obviously that destruction of the cell is going to be a major issue for that cell, right? It leads to cell death when that happens. Next, we have cell swelling. Certain viruses can make cells swell and become larger as we are like filling it up with the viral particles, and the cell is losing control of these sorts of mechanisms. This be usually accumulation of fluids or changes in the cell membrane permeability as we're having introduction of all of these spike proteins and elimination of the normal proteins that are going to regulate that sort of stuff, how much water is in or out of the cell. So it loses all control of that and can cause the cells to swell. So we have bursting as lysis, we have swelling, and then we have syncytia. So syncytium is one of them. We can have one happen. So syncytia happen whenever, like I said, the viruses cause the cells to fuse together. It's typically we're going to have the proteins of the virus on the outside of one cell, and it's engaging with the cell next to it and causing them to merge together, which is normally what the virus will want to do with the cells, but the proteins are there on the outside. So, so neighboring cells will merge. And then we have inclusion bodies. This is just going to be, I don't focus too much on the facts of this word, right? I'll just say inclusion bodies are just like little masses of leftover stuff or viral proteins or damaged parts of the cell or whatever that are building up inside of the cell, and they kind of ball up together or create these inclusion bodies, including meaning that they're including all of what viral proteins or all of the cell parts or whatever it is. And you can see these globs or areas where we have these bodies forming inside of the cell. You can see them in the microscope. So again, cytopathic effects are usually visible signs of damage in a microscope due to a viral infection, and helps us understand that viral behavior. So this was the little thing that it's not really like a mnemonic because it's not spelling out a word or anything, but sick cells develop crazy alterations. You're sick, you've got the virus, and you could be having syncytia and all this stuff. And the crazy alterations being all these changes that we see in the cells. But sick cells are going to be developing the crazy alterations. So some of our example questions here. Which of the following best describes a cytopathic effect in the context of a viral infection? So we'll go through these. A viral protein that inhibits host cell replication. So this we already talked about. This isn't something that you can see. This isn't a structural change. This isn't something that you're necessarily going to be seeing in that cell while it's being infected, right? So it wouldn't be this one because it's not something you can see in the microscope happening to the actual infected cell in that moment. So structural and functional change observed in host cells due to viral infections is pretty much exactly what we're talking about, but we'll go through the other choices as well. The process by which viruses are released from infected cells. No, I mean, that's like budding out or exocytosis. That's not what cytopathic effects are talking about here. So we're just looking at a whole bunch of different kinds of things that can have negative effects on the cell that can be seen in the microscope. The ability of viruses to integrate into the host cell genome, and that's at all. So we're really talking about this structural and functional change from the viral infection. So our answer would be B on that. Next, we have a laboratory technician observing that cells in a culture have fused to form multinucleated giant cells and exhibit several severe cytoplasmic swelling. Which of the following viral infections is most likely to be associated with these cytopathic effects based on the name? So we already see here we have giant multinucleated cells. So if they're multinucleated, we know that we have syncytia forming. And so based on that and the fact that we have the swelling of the cytoplasm of these cells as well, there's two cytopathic effects going on here. So we look through our choices and try to see which one pops out as a possibility. Can we say this about herpes simplex virus? I don't know, it's not really giving it away. The influenza virus, again, we don't know, it's not really giving it away. Human immunodeficiency virus, it's not really giving that away in its name. But respiratory syncytial virus, RSV, is giving that away. So we have syncytia formation that it's talking about here. None of the other ones talk about those things. So it's going to be that one. All right, next, I know this is a lot, but I wanted to put everything on here that we had to know about these topics. So this is talking about temperate phases, the lytic cycle, the lysogenic cycle, and lysogenic conversion, which I know that we just talked about. But briefly, just to remind everybody, temperate phages are a type of bacteriophage. Again, this is only going to be talking about bacteriophages, not eukaryotic viruses. These are only in the context of the bacteriophages. And bacteriophages are just viruses that infect bacteria. These can follow two paths, lytic or lysogenic, and it can switch between them. In the lytic cycle, it's the normal viral pathway. We're going to be creating new viruses that are going to be bursting out of the bacterial cell, which is what they always are going to do in bacteria. And then going on to the next cell and infecting them and replicating up their little, synthesizing their parts and making new viruses and bursting out whatever. So that whole process of that active replication of new virus particles is the lytic cycle, right? Think of lytic as being like lysis, if you want to. If that's not too confusing, then don't, because the next one is lysogenic cycle, and I always focus on the gen being gene. That's how I remember it. But this is the other pathway where we're going to be inserting our genome into our bacterial target host genome. So the viral genome literally gets spliced into there. And that is the lysogenic cycle. Essentially what you'll have are bacteria dividing and replicating those genes of that virus every time that they divide, but they're not being burst open by any viruses. We're not making new viruses. We're just propagating the gene that way for it. So that's how that will work. Remember lysogenic conversion. We are converting your bacteria to being more pathogenic. So we'll have an increase in pathogenesis or, disease effects, it being more, causing more obvious effects of disease if you are infected with it. So here we're talking about, and I mean in eukaryotic cells, increased pathogenesis of the bacteria in their eukaryotic host. So things like diphtheria, which we've mentioned in class, or other ones like, Clostridium botulinum for botulism, they got their toxin genes from lysogenic conversion. They're not going to cause the disease that they're known for without these genes from lysogenic conversion. They got these from viral infections originally. Now, most of these have these permanently as a result, but originally they came from phages. So that's lysogenic conversion. We've now made these bacteria pathogenic or more pathogenic by introducing these genes whenever we incorporated the viral genome in there. It carried some extra junk with it. So that helped it out. We usually see this with toxins, but we can also see it with, resistance. So yeah, but anyway, so that's the idea of it. So this one, this was just, I don't have a great explanation, but some lovely lions instantly take control, but lazy leopards lie in wait. So these are both L's, lytic and lysogenic, and that's the unfortunate aspect of it. But for lively lions, this is going to be the lytic because we're lively and we're going to be lysing open the cells, right? We're making new copies. The virus itself is very lively and doing viral things. So lively lions and lie. So like lysis. That's how I just try to think of that because I'm mostly thinking of the gen and I'm thinking of the lysogenic. Those guys, the lively lions, those are going to instantly take control, but the lazy leopards will lie in wait. And I like the lively lions instantly take control because it's LLITC for that part. So that's like lytic, right? That's kind of spells out lytic a little bit. So lively lions instantly take control. So if they're lively lions, instantly taking control, that'll be the lytic spelled out with those letters. And then if you can just remember the other ones were lazy, that's going to be the lysogenic, which is your other option. And again, the gene, where we're inserting the genome in there, They're lying in wait, right? They're waiting in order to come back out. So that's kind of what the thinking was with that. I don't know if you like it or not, but you don't have to use it if you don't like it. So which of the following best describes the main differences between the lytic and the lysogenic phases, cycles of the temperate phases? So let's go through these. So we're looking at lytic versus lysogenic of the temperate phases. Okay, so they can switch between, remember we just talked about the lively lions, right? So the lively lions, there's going to be the lytic. So that's our active multiplying, making new viruses. And the lysogenic, we're going to be inserting the genes into the genome. So now that we've established that, because we definitely remember that wonderful acronym that I made up that makes total sense. The lytic cycle involves the integration of phage DNA. Well, we can stop right there. We already know the lytic cycle, the active cycle, we're not integrating our phage DNA in that. So that integration of the phage DNA in the lytic cycle, no. So that's not A. B, the lytic cycle results in the destruction of the host cell through lysis. Okay, we're getting somewhere here. That active replication, making the viruses, if we're a bacteriophage, they always burst out of the cell, and that's called lysis. So lytic cycle, lysis, I'm seeing it. While the lysogenic cycle, remember, we see the lysogenic, we see the gene, we're integrating the genes into the genome. Yes, this is integration of the phage DNA into the host genome. We do not have immediate cell death. Yeah, because the cell is going to be replicating just like normal and replicating that genome with it as it does that. So this one's starting to make a little more sense. Lytic cycle occurs in all types of bacteria, while the lysogenic cycle only occurs in gram-positive bacteria. So in the phage infections, they're typically all going to be lytic at some point. So this is kind of true. This first part of it, lytic cycle, occurs in all types of bacteria. Yes, but the lysogenic cycle, did we say anything about it only occurring in gram-positive bacteria? We did not say that. In fact, that is not true at all. So if we didn't say that and we didn't learn that in class, that isn't, no, okay? Just cross it out. All right, the next one, the lysogenic cycle produces new phage particles immediately, whereas the lytic cycle does not produce phages. It is kind of the opposite, and this is a really watered down explanation of it, right? So lysogenic, we don't do new phage particles. We don't do it during the lysogenic, right? If you end up switching back over to make phage particles again after you've been induced, you're not in the lytic cycle. You're not in the lysogenic cycle anymore. Now you're in the lytic cycle. The lytic cycle is what makes the phages. So this is kind of the opposite of what it's supposed to be. So that leaves us with B as being our correct answer, which was what we were leaning towards anyway. So the next one, a patient presents with a bacterial infection caused by a strain of Corynebacterium diphtheriae. I know that these words look like they are horror to you, but you'll become more familiar with them. And unfortunately for you guys, when we get to Unit 4, you'll have to pronounce them. And you will not do it correctly. And I'm not going to count you off, but I do expect you to try. Because how do you think I learned to pronounce these? So it's not like I took a class on bacteria name pronunciation. I just kind of figured it out. So, but that's okay. Just, you know, be aware. Some of you might have to pronounce words like this. So Corynebacterium diphtheriae, the lab, please sit down. The lab results show that the strain is producing diphtheria toxin. So it is making a diphtheria toxin, which is kind of where it got its name, right? Clearly, I mean, it has diphtheria. So this is the disease diphtheria. And the reason it's causing the disease is because we are actually producing this toxin, diphtheria toxin, from our Corynebacterium bacteria. So which of the following explains why this strain can produce this toxin? And we talked about this just a minute ago. So that's why we can answer this question. But the bacterial strain carrying a prophage that has integrated into its genome that encodes a toxin. Well, that sounds pretty familiar. We have lysogeny that has introduced a new gene that codes for a toxin. And this leads to a process that we call lysogenic conversion, right? We talked about that. Okay, the next choice would be the bacterium has a high mutation rate that causes it to produce toxins. I mean, that we never talked about anything relating to that, at least not yet, but like typically toxin production doesn't come from mutation. You're going to make a whole new protein just from a mutation. And that's not how mutations work. You typically have a couple of nucleic acids mutating, not like a whole mass of a gene, which would be coding for a toxin. So this is kind of out of the realm of our talk of what we've learned. So not to mention it's kind of ridiculous. The bacterium has, you may not know that yet, that's okay, we're gonna get to that though. The bacterium has a high resistance to antibiotics, which leads to toxin production. What does this have to do with antibiotic resistance? No. Why would antibiotic resistance make you produce more toxin? I mean, really, if you were to ask yourself that question, like, I don't know, then the answer is, yeah, no, it doesn't. Like, that's why you don't know. It doesn't make sense because it doesn't make sense. So that's okay. Sometimes you can just knock out the ones that like sound like absurd to you, just knock them out because the other ones might make more sense. And sometimes you can just, that might be your best bet if you don't fully understand what the question is like in detail as far as that concept or whatever. If you have a vague understanding of maybe what other concepts are or what vague understanding what the overall look is, that can help you break it down too. So the last one, the infection has caused the bacteria to undergo genetic recombination, leading to toxin production. So this might be tricky for some people looking at this. They're like, well, it did have a new gene inserted into it, but the infection is what's causing the bacteria to undergo that genetic recombination. If you actually look at the wording of that, that sounds ridiculous. So you infect your bacteria, you come and infect a cell. All right, the cell's now infected. And that bacteria, because of that process of just infecting your cell, you're now undergoing genetic recombination. Genetic recombination is exactly what it sounds like, where you're going to introduce new genes into the genome. So you're going to start doing that to your genes and then start making, this is nonsense. So be just infecting a host cell, getting into your intestinal cells, let's say, as a bacterium, isn't going to cause it to, undergo genetic recombination. That's kind of an absurd thing. And besides the fact that we typically look at genetic recombination as being something that we do to cells, we introduce those genes to the cells. We learned about that in chapter 8 a little bit more. This type of question, you're not going to have this kind of complicated of a question on the exam. On the exam, there might be a lot of wordy stuff, but it's not going to have a whole bunch of stuff that is really going to have to make you break down to this almost still vague concept where you'd have to get this out of. It's not truly on the study sheet is what I'm saying, right? So you'd have to like break it down, like kind of like we talked about with the fungi not being photosynthetic. So, yeah, I'm not going to put stuff like that on the test. The test questions, they may not feel like it when you're taking the test, but I promise that they are asking overt questions about things that we actually talked about in the class directly. So remember that. If you see things that you don't recognize that we talked about in the class and you're sure that we didn't talk about it, cross it off. If you're ever not sure about something like that, there is no harm in asking me a question. The worst case scenario is going to be like, well, we talked about this in class, but you remember how we said blah, 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 about blah, blah, blah. I sometimes can give a little bit of hint if I feel sympathy. And if you're only asking like one question, if you're asking questions about every question, I might have an issue with that. But generally speaking, if you're asking just a couple questions about the exam questions, I will try to push you in the right direction, assuming that you studied. So next thing is the prions. I legit just went over this. Like I don't want to go over it again because I already went over it twice in detail. But again, these are misfolded proteins. They do not have a genome. There's no DNA or RNA that codes for this in any way, shape, or form. It's just a misfolded protein. And that misfolded protein gets near other normal proteins and causes them to misfold. It's like a more stable conformation. In fact, it's so stable that you can't even get rid of these with normal autoclaving. You can get rid of endospores with normal autoclaving. That's why we do it. But prions, no. You have to up that temperature and that pressure a lot higher than normal if you want to get rid of them. It can't even be eliminated by cooking. If you eat meat that is contaminated with these prions, even if it's completely well done and cooked and charred a little, it's probably still got the prions in it. So that's a problem, right? But this is a super stable conformation of this protein. It loses its function in the cells when it does that. So it's not the favored conformation for the cell, but it's the favored conformation for the protein, which is why the protein causes other proteins to switch that way. And I don't know if you guys have ever read this book. It's an absolutely absurd book. If you like that sort of thing, though, if you like a lot of sarcasm and cynicism in your life, which I do, it's written by Kurt Vonnegut. He wrote a lot of books like that are that way, but this particular book is called Cat's Cradle, and it's about something very similar to this. It's about water and the conformations of water, like how water molecules stack with one another with their little hydrogen bonds and their oxygens interacting, and how they favor that. Whenever you freeze water from liquid into ice, it changes the conformation of those H2Os, right? And that's how come it freezes the way that it does. They say it expands. Water is unique in that. Typically, most things, whenever they go solid, are going to shrink from the liquid size, but not water, because of how those H2O molecules will stack with each other when they freeze. The book is about having many different conformations of how the H2O molecules could stack differently, and then reach a conformation that might be the most favorable. And they found it, and it's called ice 9. So whenever you freeze water and it freezes in this specific conformation, this 9th conformation that they discovered, it is the most favorable conformation for water to want to be in ever. And so they have a crystal of it, and potentially if that crystal of ice 9 were to ever come into contact with the water of the world, it would cause all of the worlds to freeze, all of the water in the entire world to freeze instantly, because it all would want to conform to that one conformation. That is a lot like the prions. Obviously, you can tell it's going to be a very depressing book, and I quite like it. It's one of my favorite books. It's not sciencey. I don't want to make it sound sciencey. It's just people being absurd. If you like reading about people being absurd and like figuring out that they are absurd and maybe learning from their absurdity, that's kind of what his stuff is. It's very cynical and sarcastic, but yeah. And also Ice Nine, if you don't know, there's like a band out there called Ice Nine Kills. It's like a metal group that I don't know. I don't particularly care for the music, but it's like kind of horror metal if you're interested in that. But it's named after it, so that got me off track. But anyways, that's kind of how prions work. They will see that misfolded conformation, totally functionless, and say that is the way I want to be, and then turn into that, and they cannot go back. Once you've done that, that's it. There's no going back. So yeah, they will aggregate in the brain tissue and the nervous tissue and cause holes in it, and that is what leads to these diseases. So this goes into a little bit different approach, I guess, in explaining it, different words than what I typically use. If you like to read through this explanation of it, I tried to look at it, don't say look at it a different way, but just word it differently from how we've been talking about it a little, and talk about how that works. But anyways, our mnemonic for this, to help us remember about prions and what they are doing, I have prion D, and this was one that actually makes sense for once. Prion D, problems really involve only nerve damage. And so this is just talking about how the protein particles that are going to be misfolded are going to cause problems. Those prions are going to cause problems, and typically leading to only nerve damage. It really only affects the nerve, and it is going to cause permanent damage in nervous tissue like the brain and the nerve. So this is just remind you guys, this is one that's going to be causing nerve damage, and that's where we get the encephalopathy, encephalo being brain, pathy being disease. All right, so which of the following statements accurately describes prions? So prions are viruses that cause neurodegenerative diseases by introducing viral DNA into host cells. Well, prions kind of have it in their name that they're proteins, like just prions, prions, how some people will pronounce it. These are misfolded proteins, proteinaceous, infectious particles. And so these are not viruses. They're not viruses. They are acellular, but those are separate, right? Viruses and prions. In our little chart, we drew it in that other thing. So they're separate from one another. So these are not viruses. They do not have viral DNA. They don't have any DNA or RNA of any kind. So this can't be it already, even if we were going to say, okay, well, prions do cause neurodegenerative diseases. We don't have the other stuff going on. Prions are misfolded proteins that induce other normal proteins to misfold, leading to brain damage, okay? Prions are bacteria that release toxins that damage the nervous system. They are not bacteria. No. We know they're misfolded proteins. That's all they are. Prions are, okay, they're not fungi. They're not fungi, they're not bacteria, they're not viruses, they are just misfolded proteins. Already, if you just knew only that about prions, you just pick that. You don't even know that it causes even, misfolding, or that. I mean, you don't necessarily know anything, but if you knew it was a protein, you got your answer right there in B. So anyways, next one. A 55-year-old patient presents with rapid onset of dementia, coordination problems, and muscle twitching. Diagnostic tests indicate abnormal prion proteins in the brain. Which of the following diseases is most likely associated with these findings? So obviously we know where this is going. We talked already about the main disease caused by prions in humans, and that was Creutzfeldt-Jakob disease. There it is, spelled out for you in case you wanted to see it. Creutzfeldt-Jakob disease. So that's the answer. You just kind of have to know that one. But you could do, your, breaking it down of like Alzheimer's versus multiple. We didn't talk about any of these, and certainly we would have if that was important about that. So it's not those. It'll be Creutzfeldt-Jakob disease. I wanted to say about diagnostic tests for this. This isn't like testing for PCR for viruses, which usually have DNA or RNA. This isn't like testing for bacteria or fungi that also have DNA or RNA and that you can culture. This is a misfolded protein. So how do you identify a misfolded protein? That the sequence isn't bad. It's a normal protein. The amino acid sequence is normal. There's nothing mutated about it. It's just misfolded. So how do you identify that with molecular mechanisms? There are some ways to do testing on it. It's extremely rare that they ever do it, but they can take cerebrospinal fluid from people who they suspect may have a prion disease and send this to labs like in Los Alamos or something, and they can do these propagative tests. And basically they introduce that patient's sample into normal protein and then see if the normal protein misfolds. And there's ways that they can look at that in the results. But so if it does cause it to misfold, then you're positive for it. This is not a test that is available in really anywhere. It's only like in Los Alamos, New Mexico, and maybe, I don't even think the CDC does it. It's super rare because most of the time you're going to diagnose these prion diseases after death. They're 100% fatal. And so you will wait till the person dies. And during the autopsy, you will examine their brain tissue and look at the brain tissue for that spongiform effect. And that's how you diagnose it officially. Terrifying to think about, but that is typically how they would be doing it. These new tests are actually pretty new and almost no laboratories do it. And if you guys don't know, Los Alamos is government run, highly suspect facility. It's like where everybody thinks they do testing on Roswell aliens or some stupid thing like that, but that's that area. So it's a lot of classified stuff. So of course they would be having these new tests available that nobody else has. But that's it for this discussion. I know I went off tangent, but I hope that at least some of it was helpful to you guys. Of course, if you guys have any questions, you can email me. I'll be available most of the weekend. You guys know how it goes, and let me know if you need anything. I don't think anybody left any shots. No, I just want to be sure. All right, so that's it. If you guys have any questions in chat, just let me know. And otherwise, I'm gonna get off here in the 5 minutes, maybe, probably less than that. And the dogs say hi. I'm just glad that some people are finding better if I just laid on spaghetti. I'm sorry, buddy. I'm glad some people are finding some benefit from this stuff.